So with this, let me introduce the first one, Robyn. Currently a postdoc at Hopkins University in Dr. Kathleen Cullen's lab, but she will be presenting us her final project of her PhD thesis that was conducted at the School of Kinesiology at the University of British Columbia. Please, Robyn, the floor is yours.
Thank you for the introduction. So I'm Robyn Mildren, and I'm going to be presenting some of my work from my PhD thesis at UBC. So my work focused on the stretch reflex pathway. So stretch of a muscle activates muscle spindles, which send signals to the spinal cord to generate a rapid contraction of the muscle. And so I'm interested in studying this pathway in populations that have poor balance control, because we know that proprioceptive information from the ankle joint is really critical for the control of balance. So with aging, there can be progressive changes in different elements of the stretch reflex pathway. And following a stroke, there's a disruption of descending information sent from the brain to the spinal cord, and that primarily affects one side of the body. So how this pathway is typically assessed is by applying a transient stimulus to the tendon to stretch the muscle, and then recording the evoked response around 40 milliseconds later. And so I'm interested in studying this pathway during standing, so when this information is relevant to the control of balance. And one of the drawbacks of this approach is that these abrupt stimuli generate a perturbation to balance. So we've previously developed this method that involves delivering a continuous low-level noisy vibration to the Achilles tendon, and then looking at its linear correlation with ongoing EMG. So this stimulus contains frequencies between 10 and 115 hertz, which are frequencies that muscle spindles can encode and are generated naturally during normal activities. And this allows us to probe responses across a relevant frequency bandwidth without noticeably perturbing standing balance, and also with relatively short trial durations. And so using this technique, the aim of this study was to examine how healthy aging and chronic stroke influence lower limb muscle responses to proprioceptive stimuli during standing. And we also wanted to explore potential mechanisms that might underlie these changes and whether they're related to postural control. So here we tested 54 healthy adults across a broad age range, and that allowed us to examine changes across the adult lifespan, and 8 individuals with chronic hemiparetic stroke. And we examined soleus responses, postural control, and mechanical properties of the muscle tendon unit. So this is showing the cross correlation between the vibration and EMG. So as you lag the EMG around 40 milliseconds relative to the stimulus, those signals are correlated. And this is plotting the peak-to-peak cross correlation as a function of age. So you can see the soleus response amplitude declines with age. So here I've grouped individuals into young, middle-aged, and older groups. And this is showing coherence, which is the strength of the linear correlation between the vibration and EMG across frequencies. So coherence became weaker with age and also spanned a narrower frequency bandwidth. So gain is the muscle output relative to the input. And similarly, we found that gain also became lower with age. So to examine whether changes in mechanical properties of the muscle tendon unit could mediate some of the changes we saw in soleus responses, I examined mechanical admittance, which is the relationship between the force and the velocity of the probe against the tendon. And we found minimal age-related differences in mechanical properties. So overall, these progressive changes in the characteristics of the soleus response to proprioceptive stimuli is likely not driven by changes in mechanical properties, and also seems to reflect the progressive changes that have been described in the morphology of muscle spindles across the lifespan in animals. And we also found that a combination of age and soleus response amplitude could predict postural sway frequency, but the soleus responses alone had minimal relationships with postural control measures. So overall, I think that insight into how proprioceptive information changes across the lifespan could have some implications for prehabilitation with aging in order to reduce falls. So next, I examined individuals with chronic hemiparetic stroke, and this is showing center of pressure traces under the more affected limb and the less affected limb during quiet standing. So both the frequency and amplitude of adjustments was lower on the affected limb, meaning that that limb is contributing less to the control of balance. And so unlike with aging, we did see differences in mechanical admittance, where the affected side tended to show lower admittance, particularly at these higher frequencies, meaning that the tissue is impeding the motion of the probe more. And these individuals with altered admittance also tended to show larger soleus responses on the affected side. So here there's potentially some relationships between postural control, muscle tendon mechanical properties, and soleus responses to proprioceptive stimuli. So I think that potentially assessing and trying to mitigate some of these frequency dependent changes in mechanical properties could have some implications for improving postural control post-stroke. Thank you for listening.
Thank you very much. Thanks. So please type away your questions in the Q&A section, but I might get started because I don't work with stroke patients, but I do work with dementia patients. So I was wondering, my mind immediately went and wonder whether you have tried or are thinking of trying it with Parkinson's or other kind of non-healthy aging populations and compare the results.
We've definitely thought about it. The one population that would be really interesting to test is peripheral neuropathy. Parkinson's would also be interesting. I've actually tested one individual with Parkinson's. The problem with the reflex responses is they're quite variable. So with one individual, it's really, really hard to tell. Peripheral neuropathy is like something that would be very interesting because you have this sort of distal to proximal loss of sensory information, and how that really affects spindles is a little bit less clear than how it affects skin receptors in the periphery. So that's something that would be really interesting to test. And did you say, what other population did you mention?
Oh yeah, I work with neurodegenerative patients, so I just thought in general, not healthy aging, so still aging, but with some, it's incredibly interesting. You mentioned the variability, so, sorry, we have questions from, and I'm not gonna monopolize. Ruyan Wei is asking, why the less effect leg have more movement trajectory?
Yeah. For the center of pressure traces. Yeah. So the affected side will have less movement. Overall, if you merge the force plates, the individuals that have chronic stroke will actually have more sway altogether if you sum it all up. But when they're standing and you're just looking at each limb independently, they're basically using that affected side more as a passive brace. I mean, it's maybe contributing a little bit to balance control, but I think most of the adjustments are being made.
Okay.
Oh, sorry. I have those. Yeah. So most of the adjustments are being made on the less affected side. Does that make sense?
It does. I think Kami as well has a question.
Yeah, thank you for your presentation. On this slide, I was actually wondering what it means when the affected leg has variance in the admittance compared to the less affected leg in the high frequency. Like, what do the frequencies mean physiologically?
So it's a little bit, I don't have a great answer to that because to me, this is all fairly high frequency. What you could pull out is where you have peaks is where you might have some resonance, which is something that I was trying to look for. And then that resonant frequency should be related to stiffness. So stiffer structure, higher resonant frequency. And so you do see those differences. But overall, I think I'd have to look into this in more detail in terms of both like what the tendon is doing and what the muscle is doing to come up with this pattern when there's sort of like an inflection, sometimes around 50 hertz. because this is looking at like the combination of the mechanics of the tendon along with the muscle. And there's some evidence that the tendon's actually becoming slightly more compliant while the muscle is actually becoming more rigid. And so how they're really interacting is something that we really need to look into further, and potentially with some imaging like ultrasound imaging.
Right. We have a question from Carly Sombrick, asking whether you would expect that the stroke deficit would be related to the extent of the gait recovery.
Gait recovery, you said?
Yeah.
Yeah, sorry, the extent of the
extent of, would you expect that the stroke deficit would be related to their extent of gait recovery?
So which deficit, the change in admittance, or?
Yeah, this is how I'm interpreting the question.
Yeah, that's a good question. We did do a lot of clinical tests, Fugle-Meyer, timed up and go, and Shadok-McMaster, some other tests. So I like the timed up and go test. I should look into that in a little more detail, but I think definitely there are relationships between like motor control, gait, and the mechanical properties. That seemed to come out fairly strongly. So these individuals with this like sort of blunted admittance at higher frequencies really had like poor measures on tug test, which involves gait as well as transitions, and also like more impaired motor recovery on the Fugle-Meyer as well.
Yeah, I believe that the next question from David Granite is always in this line. So he's wondering whether the changes in admittance in the stroke population are results of differences in tonic activation of the muscle. Like if there's...
I don't think so. I did also record EMG, and EMG tends to be lower on the affected side during standing, even if I try to get them to stand symmetrically with equal forces under both limb. So I think that I think almost like higher muscle activity would stiffen that muscle tendon unit and cause that sort of blunted admittance, but it's actually the opposite. There's lower activity on that affected side.
Right. We have another question on the stroke patients because it's so interesting, the result. Nick Kitchen is saying, well, first of all, thanks for the talk. And wondering how old were the patients, and could their age explain some of the differences, some of the deficits you observed?
Yeah, so the beauty of this project was I had 54 healthy adults to age match to. So I have age match controls that are all within 2 years. So there's clearly differences both between affected and less affected side, and also between stroke and control, like the affected side of stroke and healthy controls. And within healthy controls, there's no differences between the right and left leg in any measures.
Nice. Amna Lawrence is kind of asking a following up question on possible alternative populations, and wondering whether you think your study would be applicable in patients with multiple sclerosis.
Yeah, I'm actually really interested in that disorder, but potentially some different studies. Like I'm really interested in how that affects transmission through, how MS affects transmission through spindle afferents, because I think potentially there could be some changes in where spindles branch and branch point failure of action potentials through that branching, because muscle spindles branch to contact almost every single motor neuron in the motor pool of the muscle that they've come from. And so I think there could be potentially quite large changes with MS, and that's something I'd love to look at.
Very interesting paradigm that it sounds like you're going to have to keep on working on this and applying to numerous different populations. So thank you very much, Robyn. Keep an eye on the Q&A because people might still write you.